Hi everyone, it's Dr. LeBlanc with Unit 7D, Repair. Our learning goals are to understand the importance of DNA repair mechanisms for survival, to understand the 5 different DNA repair mechanisms: direct repair, base excision repair, nucleotide excision repair, mismatch repair, double-stranded break repair, and to understand translesion DNA synthesis as a mechanism of coping with DNA damage. In September of 2015, the Nobel Prize in Chemistry was awarded to Thomas Lindahl, Paul Modric, and Aziz Sankar for having mapped at a molecular level how cells repair damaged DNA and safeguard the genetic information. Their work, according to the Nobel Prize Committee, has provided fundamental knowledge of how a living cell functions and is, for instance, used for the development of new cancer treatments. To review, mutations are heritable changes in the genetic material. Mutations provide allelic variation and are the foundations for evolutionary change. However, they are usually detrimental. Therefore, cells have evolved many and overlapping repair mechanisms. DNA repair is a multi-step process. An irregularity in the DNA structure is detected. In most mechanisms, the abnormal DNA is removed and normal DNA is synthesized. However, we're going to discuss a DNA repair mechanism called direct repair that doesn't remove any nucleotides. 2 different types of direct repair: one to repair thymine dimers and another to repair incorrectly methylated base. First of all, ultraviolet radiation can covalently link 2 thymine residues that are next to each other on a strand of DNA. This is a problem for the DNA replication machinery. DNA replicase cannot synthesize new DNA past a thymine dimer. So a mechanism of direct repair is to have an enzyme called DNA photolyase simply break the bonds that were created by the ultraviolet radiation, and the normal structure of the thymines is restored. For direct repair of a methylated base, here's a O6 methylguanine. It has a methyl group here where it's not supposed to be. Alkylyl transferase can come in and simply remove the methyl group and restore the normal guanine base structure. So these are 2 examples of direct repair. The base excision and nucleotide excision repair, however, are extremely important for many other types of damage to DNA. In the early 1970s, scientists believed that DNA was very, very stable. They had been working with DNA and RNA. They knew how unstable RNA was, so they thought DNA was really very, very stable. However, Thomas Lindahl demonstrated that DNA decays at a rate that ought to have made the development of life on Earth impossible. This insight led him to look for and discover molecular machinery called base excision repair, which constantly counteracts the collapse of the DNA molecules in our cells. So, base excision repair. Depending on whether a purine or a pyrimidine is removed, this creates an apurinic or an apyrimidinic site, and it has to be that's part of the DNA repair mechanism. In the subsequent slides, we're only going to refer to the apyrimidinic site, but in actual fact, this repair mechanism works for either apyrimidinic or apurinic sites. So what happens first? First, the N-glycosylase recognizes an abnormal base in the DNA. Here's our U, okay? It's not supposed to be there. It's supposed to be a T opposite the A. So N-glycosylase cleaves the bond between the base and the sugar. So that's the very first thing. We just removed the base, not the whole nucleotide. Okay, that leaves an apyrimidinic site in this case. but remember it also works for apurinic sites. AP endonuclease recognizes the missing base and cleaves the DNA backbone right here on the 5' side of the missing base. Now in E. coli, DNA polymerase I uses, remember DNA polymerase I? It uses its 5', 3' exonuclease activity to chew back, and then it fills it in at the same time with normal DNA, and then DNA ligase will seal up the region, and you will have a newly synthesized, completely newly repaired piece of DNA. What about in eukaryotes, such as humans? Humans are eukaryotes. A different DNA polymerase called DNA polymerase beta can remove the apyrimidinic site and replace it with the correct nucleotide. DNA ligase seals the region. Okay. Also in eukaryotes, such as humans, there's a different DNA polymerase, delta or epsilon, can synthesize a short segment of DNA and generate a flap. Okay, so here's our new sequence of DNA. We've got a little flap here. Remember flap endonuclease? That flap endonuclease can remove this little piece of DNA, and DNA ligase will seal it up. Okay, so that's base excision repair. Remember, we started out with a apurinic or apyrimidinic site after an incorrect base was recognized in the DNA. Another type of repair called nuclear excision repair removes an entire nucleotide. Aziz Sankar mapped nucleotide excision repair, the mechanism that cells use primarily to repair UV damage to DNA. We have the direct repair that we talked about, but that's not enough. We need more than one repair mechanism to fix all the damage that our DNA incurs. Nucleotide excision repair is the mechanism that cells use to repair UV damage to DNA. There are people who are born with defects in this repair system, and they will develop skin cancer if exposed to sunlight. The cell also uses nucleotide excision repair to correct defects caused by mutagenic substances, among other things. So let's go through the steps of nucleotide excision repair. Here's a thymine dimer in DNA. It has not been repaired by photolyase. It's still there, and if it's still there, the cell is going to die. It will not be able to replicate its DNA. So there's a surveillance complex called UVRA, UVRB complex for ultraviolet radiation. A and B are the two different proteins, and scans the DNA until they find a thymine dimer. Okay, once they do that, UVRA is released from the complex, UVRB binds, and another protein, UVRC, this big long protein, comes and binds to UVRB. This protein makes cuts in the sugar phosphate backbone of the DNA, and this little piece of DNA is removed by UVRD, which is a helicase. Okay? Then DNA polymerase I again comes in and in a 5' to 3' direction fills in the missing nucleotides, and then ligase will seal up the final sugar phosphate gap. Defects in the nucleotide excision repair components in humans cause human genetic diseases. This individual, you can see all the freckling on his face and in the region near around the top where his shirt might have been open. Those freckles are caused by the defects in the nucleotide excision repair. The disease is called xeroderma pigmentosum. There's a couple of other diseases called Cockayne syndrome and PIBIDS. Xeroderma pigmentosum can be caused by defects in 7 different nucleotide excision repair genes. We have a few more than E. coli does. And defects in any of those genes can cause xeroderma pigmentosum. Finally, the Nobel Prize was awarded to Paul Modrich for his work on mismatch repair. This mechanism reduces the error frequency that DNA replication might have left behind by about 1,000-fold. If you have mutations in this repair mechanism, you end up with congenital defects that can cause a hereditary variant of colon cancer. So mismatch repair, obviously, as the name implies, a mismatch is when two nucleotides opposite each other are incorrect. So there are several proteins that are involved in this, Mut-S, Mut-L, Mut-H, but what I want you to take particular note of is that we need to remember that directly after DNA replication, okay, immediately after DNA replication, the DNA is in a hemimethylated state. The methyl groups that exist on the parental strand have not yet been duplicated on the newly synthesized strand, and the mismatch repair mechanism takes advantage of this discrepancy and has designed a system where you can recognize the parental DNA and discriminate it from the newly synthesized DNA, which is very important when you want to remove a mismatch. So here's our first step in mismatch repair. We have our parental strand here in dark purple and our newly synthesized strand in light purple, and a methyl group attached to a parental strand. There are several proteins that come in and recognize this situation. Okay? First of all, the mismatch is recognized by Mut-S. Mut-H recognizes the hemimethylated DNA, and Mut-L acts as a go-between. So the Mut-S protein finds the mismatch. The Mut-S, Mut-L complex bind to Mut-H, which is already bound to the hemimethylated sequence. Mut-H makes a cut in the DNA in the non-methylated strand. The non-methylated strand is the newly synthesized strand. Mut-H separates the DNA strand at the cleavage sites, and an exonuclease digests the non-methylated strand just beyond the base mismatch. So you makes the cut here, and you chew away this DNA until you get past the mismatch, okay, right here, and then DNA polymerase will come in again and synthesize new DNA and put the new base in the correct base pairing, a C opposite the G instead of the T. Okay, I'm skipping over number 4. We're going to talk about that in a subsequent lecture. Non-homologous end joining is a desperate act to respond to a very catastrophic event that DNA might incur. Okay, a double-stranded DNA break, and DNA is completely devastating. If the cell does not fix this, it will die. So this mechanism doesn't attempt to fill in any missing DNA or fix any sequences of DNA. It's just there to get these two ends back together. So the first thing that happens is proteins bind to this end, this broken parts of the DNA. So more proteins come in and bridge the gap here. They stick to each other. Okay. And then this processing that occurs, a few nucleotides are removed so that you have a little single-stranded region here, and some the gap filling occurs, and ligase seals up the ends. Okay. There's a lot of other steps in here that I'm not going over, but basically what the concept that I want you to understand is that there are proteins that find each other and then seal up this end without attempting to fix any missing or nucleotides that might be there. There's no way to do that. Okay, so it's really a desperate measures to respond to a catastrophic event. There is another mechanism that we're going to talk about, like I said, in a subsequent lecture. Okay, talk about desperation. Translesion DNA synthesis by DNA polymerase Y is the most desperate mechanism of all, and we know that it is something that occurs frequently. The Y family of DNA polymerases have examples in all species from yeast all the way up to humans. What does translesion DNA synthesis involve? Well, here we have a section of the DNA that's damaged for whatever reason. And here's the replication machinery. It bumps into this DNA damage, and it cannot synthesize any DNA across it. It's there. It's not going to be able to do its job. So it leaves, and a new DNA polymerase called the translesion DNA synthesis polymerase comes in, and it just throws in any old extra nucleotides it can until it bypasses the damage and continues on, and then the regular replicase comes back on. So the translesion DNA polymerase synthesizes something past this damaged DNA, but it does not remove the damage. The damage remains in the DNA molecule. Clearly, it's a very important survival mechanism, otherwise it would not have been preserved throughout evolution, but it has. So thank your DNA polymerase Ys for your current position on planet Earth. Here are the 5 different DNA repair mechanisms and a mechanism of coping with a DNA damage that doesn't actually repair it. Direct repair, we talked about, directly repairing thymine dimers by cleaving that bond. Base excision repair and nucleotide excision repair, either repairing an incorrect base or removing a nucleotide that's incorrectly paired, or a thymine dimer. Mismatch repair, using hemimethylated DNA as a cue as to which base you're going to remove. Homologous recombination repair, we're going to talk about that later, and non-homologous end joining, when you have a double-stranded DNA break and you need to seal those 2 ends back together.